Imagine a world vastly different from our own, a planet still in its turbulent infancy. Over 4 billion years ago, Earth was an utterly alien and inhospitable place. There were no sprawling forests, no vast grasslands teeming with creatures, and no blue skies filled with the songs of birds. The air itself was a toxic mixture, devoid of the oxygen we depend on. The landscape was a chaotic canvas painted with violence and extreme forces. Towering volcanoes constantly erupted, spewing molten rock and thick, choking gases into the atmosphere, creating a permanent, gloomy twilight across the planet's surface. This was a world not yet sculpted for biology, a realm governed purely by the raw power of geology and chemistry. The oceans that covered much of this early Earth were not the placid, life-filled bodies of water we know today. They were dark, murky, and likely acidic, a chemical soup stirred by relentless volcanic heat from below and bombarded by cosmic radiation from above. Violent planet-spanning storms raged for centuries, their fury unleashing torrents of rain and generating colossal lightning strikes that illuminated the chaotic scenes in brief, blinding flashes. The very ground was unstable, with continents just beginning to form and shift, causing constant earthquakes and further volcanic activity. In this primordial theater of chaos, there were no signs of life, no humans, no animals, not even the simplest bacteria had yet made their appearance on this dramatic stage. It is from this seemingly hellish and sterile environment that one of the greatest stories in the history of the universe begins. The question of how something as complex, intricate, and organized as life could emerge from such utter disarray is perhaps the most profound puzzle science has ever sought to solve. The transition from non-living matter to the first living organism was not an instantaneous event or a miraculous intervention. Instead, it was a gradual, step-by-step process, a long and intricate journey guided by the fundamental laws of physics and chemistry. The very elements forged in the hearts of dying stars, which made up the rocks, the water, and the atmosphere of this young planet, held the potential for something extraordinary. This narrative is not one of mythology or divine creation, but a story rooted in scientific inquiry and discovery. It is the story of how, against all odds, life found a way to begin. This journey takes us from the simplest of atoms, swirling in the primordial soup, to the formation of complex molecules, the building blocks of all living things. It is a testament to the creative power of nature, demonstrating how a chaotic, non-living world could give rise to order, complexity, and eventually consciousness. Welcome to the incredible, mind-bending story of our deepest origins, the epic tale of how life began, tracing its path from simple atoms to the first primitive cells, and ultimately, to us. For millennia, humanity has looked at the world and asked a fundamental question: where did all of this come from? We observe life in its breathtaking diversity, from the smallest microbe to the largest whale, and we naturally wonder about its ultimate source. Throughout most of human history, this question was answered through stories of gods, spirits, or cosmic forces that breathed life into an inanimate world. The concept that life could only ever arise from pre-existing life, a principle known as abiogenesis, seemed intuitive and self-evident. A seed grows into a plant, an egg hatches into a bird, and a baby is born from its mother. This cycle of life begetting life is all we have ever directly observed in our daily experience. This observation, however, leads to an inescapable logical paradox when we trace our origins back in time. If all life must come from other life, then how did the very first life form begin? There had to be a point in the distant past when no life existed at all. The planet was barren, a sterile rock orbiting a young sun. This presents the ultimate chicken and egg problem of biology. Before the first living cell, there was nothing alive to produce it. Therefore, at some moment in Earth's history, the established rule must have been broken. Life had to emerge from something that was not alive, a process that seems to defy our everyday understanding of the world. This is where the scientific field of abiogenesis enters the picture, offering a rational framework for this monumental transition. Abiogenesis is the scientific theory that life arose naturally from non-living matter through a gradual process of increasing chemical complexity. It is not a theory about sudden, spontaneous generation of complex organisms from mud or dust, an old idea long debunked. Instead, it proposes that under the unique conditions of early Earth, simple inorganic molecules could have reacted to form the basic organic building blocks of life. These building blocks then assembled into more complex structures, eventually leading to a self-replicating system that we would recognize as being alive. The journey from non-life to life was not a single event but a long, multi-stage process. It began with the humble atom, the fundamental unit of matter. These atoms, forged in the furnaces of stars and scattered across the cosmos, bonded together in the chemical crucible of early Earth to form simple molecules like water, methane, and ammonia. Under the influence of energy from lightning, volcanic heat, and ultraviolet radiation, these simple molecules underwent further reactions, forming more complex organic compounds such as amino acids and nucleotides. These were not yet life, but they were the essential components, the Lego bricks from which life could eventually be constructed. They were the first crucial steps on a long road toward biology. The core idea of abiogenesis rests on the premise that life is, at its most fundamental level, a form of highly organized chemistry. The incredible functions that define living organisms, metabolism, growth, reproduction, and evolution, are all driven by complex chemical reactions. Therefore, the origin of life must be sought in the origin of this specific type of chemistry. The challenge for scientists is to understand how a collection of simple, non-living chemicals could have spontaneously organized themselves into a system that could sustain itself, replicate, and evolve over time. This is not a matter of pure chance but of chemical necessity, where certain molecular interactions are favored under specific environmental conditions. The key players in this ancient chemical drama were molecules like amino acids and nucleotides. Amino acids are the molecular subunits that link together in long chains to form proteins. Proteins are the workhorses of the cell, acting as enzymes to speed up chemical reactions, providing structural support, and performing a vast array of other vital tasks. Nucleotides, on the other hand, are the building blocks of nucleic acids like RNA, ribonucleic acid, and DNA, deoxyribonucleic acid. These molecules are the information carriers of life, holding the genetic blueprint that instructs a cell on how to build its components and how to replicate itself. The formation of these critical molecules from the simpler chemicals present on early Earth was the essential first act. Once these building blocks were present, the next crucial step was their assembly into more complex structures. Scientists believed that surfaces like clay minerals or shallow hydrothermal vents could have acted as catalysts, providing a scaffold where these simple molecules could concentrate and link together into long polymers. Amino acids joined to become proteins, and nucleotides joined to become RNA. The emergence of RNA is considered a particularly pivotal moment in this story, leading to the RNA world hypothesis. This theory suggests that RNA, not DNA, was the original genetic material. Crucially, RNA is not just an information carrier. Some RNA molecules, called ribozymes, can also act as enzymes, catalyzing their own replication. This dual ability of RNA to both store information and perform chemical work makes it a prime candidate for the molecule that bridged the gap between non-living chemistry and life. In this RNA world, strands of RNA would have existed capable of making copies of themselves, albeit imperfectly. These imperfections, or mutations, would lead to variations among the RNA molecules. Some variations might be better at replicating or more stable than others, leading to a form of molecular natural selection. The RNA strands that were most successful at copying themselves would become more common. This was not yet life as we know it, confined within a cell, but it was the dawn of evolution, the driving force that would shape all life to come. We are surrounded by a world where life consistently arises from other life. A mighty oak tree grows from a tiny acorn, a lion cub is born from its mother, and even the smallest bacterium divides to create a new generation. This principle, known as biogenesis, feels like a fundamental and unbreakable law of nature. It's what we see every day, reinforcing the idea that the intricate complexity of a living organism could only ever be produced by another living organism. This observation, however, forces us to confront a profound paradox when we consider the very beginning of our planet's story. If we trace the lineage of life backward in time, we must inevitably arrive at a point where there was no preceding life to pass on its spark. This is the ultimate biological conundrum. The scientific concept that addresses this very problem is called abiogenesis. The term itself is derived from Greek roots, a meaning not, bio meaning life, and genesis meaning origin or beginning. Put simply, abiogenesis is the theory that life on Earth arose from non-living matter through a sequence of natural, physical, and chemical processes. It is a crucial distinction from the outdated notion of spontaneous generation, which imagined that fully formed organisms like mice could suddenly appear from hay or maggots from rotting meat. Abiogenesis does not propose such magical leaps. Instead, it describes a slow, gradual journey of increasing chemical complexity, unfolding over millions of years on a young and volatile planet. The core idea of abiogenesis is that under the correct set of environmental conditions, the emergence of life is not a miraculous accident but a probable, almost inevitable outcome. It posits that the laws of chemistry and physics, which govern the entire universe, are sufficient to explain the transition from a collection of simple, inanimate chemicals to a structured, self-sustaining, and replicating system. The theory does not claim to have all the answers yet, as the precise sequence of events is still a subject of intense scientific investigation. However, it provides a rational and testable framework for understanding our deepest origins, moving the question from the realm of philosophy or faith into the domain of experimental science. This journey from non-life to life was not a single defined event, but rather a long and intricate pathway with many intermediate steps. It's a story that begins with the most basic constituents of our planet, atoms and simple inorganic molecules present in the atmosphere and oceans of early Earth. Through the input of energy from sources like lightning, volcanic heat, and the sun's ultraviolet radiation, these simple precursors were forged into the fundamental building blocks of biology. This was not yet life, but it was the critical first stage. It was the moment the planet's raw materials began to assemble into something with the potential to become much, much more. So, to comprehend how life could emerge from a non-living world, we must first understand what life is made of at its most fundamental chemical level. Every living thing on Earth, from the bacteria in the soil to the cells in your own body, is constructed from a specific set of organic molecules. These are the essential components, the universal Lego bricks that nature uses to build organisms. Chief among these are the amino acids. There are 20 common types of amino acids that our cells use to build proteins. These proteins are the true workhorses of life, acting as microscopic machines that carry out nearly all the functions within a cell, from digesting food to contracting muscles to building cellular structures. Another critical class of molecules are the nucleotides. These are the individual units that link together to form the long chains of nucleic acids, DNA and RNA. Deoxyribonucleic acid, or DNA, is the famous double helix that contains the genetic blueprint for an organism. It is the master instruction manual holding all the information needed to build and operate a living being. Ribonucleic acid, or RNA, is a close relative of DNA and plays several vital roles, including carrying the instructions from DNA to the cell's protein-building machinery. The creation of these key molecules, amino acids and nucleotides, from the simpler chemicals thought to be present on early Earth represents the very first and perhaps most crucial hurdle that had to be overcome on the path to life. A third essential group of molecules are the lipids, more commonly known as fats. These molecules have a unique property. One end is attracted to water, and the other end is repelled by it. When placed in water, lipids spontaneously arrange themselves into spheres or sheets, forming membranes. This behavior is absolutely critical because these membranes can create enclosed compartments, separating an internal environment from the outside world. This is the basic structure of a cell membrane, the boundary that defines a cell and contains all of its internal chemistry. Without lipids to form these containers, the complex molecules of life would simply float away and disperse in the primordial ocean. The challenge for abiogenesis, then, is to explain how these three types of molecules, amino acids, nucleotides, and lipids, could have formed and accumulated in the harsh environment of early Earth. The atmosphere was a toxic mix of methane, ammonia, water vapor, and hydrogen with very little oxygen. The planet was bombarded by intense ultraviolet radiation from the sun and rocked by constant volcanic activity and electrical storms. While this sounds like a destructive environment, scientists now see it as a powerful chemical reactor. The immense energy from lightning and UV light could have provided the spark needed to break apart the simple atmospheric gases and reassemble them into the more complex building blocks of life. The formation of individual building blocks like amino acids and nucleotides was a monumental step, but it was only the beginning of the story. A pile of loose bricks is not a house, and a collection of free-floating molecules is not life. The next critical stage in the journey of abiogenesis was polymerization, the process of linking these simple molecular units, or monomers, into long complex chains called polymers. This is how nature builds the large, functional molecules essential for life. Amino acids must be joined together in specific sequences to create proteins, and nucleotides must be linked to form the informational chains of RNA and DNA. This process of creating long, stable polymers from a dilute soup of monomers is a significant chemical challenge. In the vast and watery expanse of the early oceans, simple monomers would have been far too diluted to link up on their own. The chemical reaction that joins them, known as dehydration synthesis, actually releases a water molecule. According to the laws of chemistry, such reactions are highly unfavorable in a water-rich environment like the open ocean. Therefore, scientists have proposed several plausible scenarios where these polymerization reactions could have occurred. One leading hypothesis involves shallow, evaporating pools or lagoons. As water in a warm little pond, as Charles Darwin once mused, evaporated, the concentration of monomers would increase dramatically, making it much more likely for them to collide and link together into long polymer chains. Another compelling location for polymerization is at the site of deep-sea hydrothermal vents. These are fissures on the ocean floor where superheated, mineral-rich water from Earth's interior gushes out into the cold ocean. The porous, sponge-like mineral structures of these vents could have acted as a perfect chemical factory. The tiny compartments within the rock could trap and concentrate the monomer building blocks, while the metallic minerals embedded in the rock surfaces like iron and nickel sulfides could have acted as catalysts, speeding up the chemical reactions that link them into polymers. The flow of chemicals and the temperature gradients at these vents would have provided a continuous source of energy and materials to drive this process forward. A third possibility involves the surfaces of clay minerals. Clays are made up of microscopic, layered sheets of silicate minerals. These flat surfaces have a slight electrical charge, which can attract and bind organic molecules from the surrounding water. Once held in place on the clay surface, the monomers would be neatly arranged and concentrated, making it far easier for them to polymerize into chains like proteins and nucleic acids. Experiments in the laboratory have shown that clays like Miller-Urey can indeed catalyze the formation of long RNA strands from individual nucleotides. Whichever of these environments was the primary stage, the formation of complex polymers was the moment when chemistry began to create structure and information, setting the scene for the next great leap. Once the primordial soup contained complex polymers like proteins and nucleic acids, a new problem arose. How did these molecules begin to work together? In modern life, DNA stores the genetic information, RNA acts as a messenger to carry that information, and proteins do the actual work of building and maintaining the cell. This system is incredibly complex and interdependent. DNA cannot be replicated without protein enzymes, and proteins cannot be built without the instructions from DNA, delivered by RNA. This creates another chicken and egg paradox. Which came first, the information or the function? The RNA world hypothesis offers an elegant solution to this puzzle. This hypothesis proposes that the earliest forms of life were based not on DNA and proteins, but on RNA alone. This idea, first proposed in the 1960s and gaining significant traction with later discoveries, hinges on a remarkable dual capability of the RNA molecule. Like DNA, RNA can store and transmit genetic information within its sequence of nucleotides. However, unlike DNA, some RNA molecules can also fold into complex three-dimensional shapes and act as enzymes catalyzing chemical reactions, much like proteins do. These catalytic RNA molecules are known as ribozymes. This discovery was a game-changer, as it meant that a single type of molecule could, in principle, perform both of the essential tasks required for life: storing information and carrying out metabolic functions. In this hypothetical RNA world, the early oceans would have been populated by a diverse collection of self-replicating RNA strands. These ribozymes would have been capable of making copies of themselves, using the free-floating nucleotides in the environment as raw material. The replication process would not have been perfect, leading to frequent errors or mutations. These mutations would create new variations of RNA molecules with slightly different sequences and therefore different properties. Some might replicate faster, others might be more stable and last longer, and some might even develop new catalytic abilities, such as being able to synthesize other useful molecules. This was the beginning of evolution at the molecular level. This process of molecular competition and natural selection would have gradually favored the more efficient and versatile RNA molecules. Over vast stretches of time, this could have led to the development of increasingly complex systems of interacting RNA molecules, all contained within protective lipid membranes, forming the first protocells. Eventually, some of these RNA systems may have discovered how to synthesize proteins, which are generally more efficient catalysts than ribozymes. At a later stage, the genetic information may have been transferred to the more stable DNA molecule for long-term storage, leaving RNA to perform its modern-day roles as a messenger and regulator. The RNA world thus provides a plausible and powerful bridge, connecting the world of simple, non-living chemistry to the complex DNA and protein-based cellular life we know today. To understand the origins of life, we must first travel back in time, over 4 billion years, to a version of Earth that would be utterly unrecognizable to us today. This was the Hadean Eon, a name fittingly derived from Hades, the underworld of Greek mythology. The planet was a violent, chaotic, and hostile world, still recovering from its cataclysmic formation. The surface was not a place of serene landscapes but a dynamic and treacherous terrain, shaped by relentless geological forces. Massive volcanoes dominated the horizon, their peaks glowing with molten lava. They constantly spewed forth immense clouds of gas and ash, blanketing the planet in a thick, perpetual twilight, and releasing tremendous heat from the planet's molten interior into the environment. The very ground beneath one's feet would have been unstable and in constant flux. The process of plate tectonics, which today shapes our continents and oceans over geological timescales, was in its infancy and far more vigorous. The crust was thinner, hotter, and more prone to fracturing, leading to constant earthquakes that would have dwarfed any we experience today. Entire landscapes could be created and destroyed in geological instants. Furthermore, the early solar system was a much more crowded and dangerous place. Earth was subjected to a continuous and heavy bombardment by asteroids and comets, leftovers from the formation of the planets. These impacts, some from objects hundreds of kilometers in diameter, would have repeatedly sterilized the planet's surface, vaporizing any fledgling oceans and melting the crust. This period, known as the Late Heavy Bombardment, was an era of unimaginable violence. Each major impact event would have acted as a global reset button, releasing enough energy to boil the oceans and transform the planet into a temporary steam world. It seems counterintuitive that life could have begun in such a hellish environment. However, this very chaos may have been a critical ingredient. The constant delivery of new materials from asteroids and comets, including water and organic compounds, would have continuously enriched the planet's chemical inventory. The immense heat and pressure from impacts could have also driven unique chemical reactions, contributing to the synthesis of more complex molecules necessary for life's emergence. It was a world teetering on the edge of creation and destruction. The surface was a raw, elemental battleground where geology and astronomy collided with incredible force. There were no green forests, no blue skies, and certainly no signs of life. The planet was a sterile, rocky sphere shrouded in a toxic haze, a far cry from the vibrant living world we inhabit. Yet it was within this very furnace of primordial chaos that the stage was being set. The extreme conditions, while seemingly inhospitable, provided the raw materials and the powerful energy sources that would transform a barren rock into a chemical laboratory on a planetary scale, making it the perfect, if violent, cradle for life. The sky of early Earth was not the familiar blue canopy we see today. It was a thick, oppressive, and alien shroud, composed of gases that would be lethal to almost all modern life. The most significant difference was the complete lack of free oxygen. Oxygen, the gas that sustains us, is largely a byproduct of life itself, produced through photosynthesis by plants and cyanobacteria. Before life emerged, the atmosphere was what scientists call a reducing environment. It was dominated by gases released from volcanic activity, vast quantities of water vapor, carbon dioxide, and nitrogen, mixed with significant amounts of hydrogen, methane, and ammonia. This specific chemical cocktail was of profound importance for the origin of life. This dense atmosphere would have created an intense greenhouse effect, trapping heat and keeping the planet's surface scorching hot, even though the young sun was significantly fainter than it is today. The sky would have likely appeared a hazy reddish-brown or orange due to the methane smog, rather than blue. With no protective ozone layer which is formed from oxygen, the surface of the planet was relentlessly bombarded by high-energy ultraviolet radiation from the sun. This radiation, while dangerous to modern life, would have been a powerful energy source, capable of breaking apart the simple molecules in the atmosphere and driving the chemical reactions needed to form more complex organic compounds, the very building blocks of biology. Beneath this toxic sky, the first oceans began to form as the planet gradually cooled enough for the immense quantities of water vapor in the atmosphere to condense and fall as rain. This was no gentle shower, it was a deluge that may have lasted for millions of years, slowly filling the vast basins of the planet's surface. These primordial oceans were not the clear, salty bodies of water we know. They would have been a hot, acidic and murky soup, often described as a dilute broth. They were rich in dissolved minerals, particularly iron, which would have given the water a greenish hue. The constant runoff from the barren volcanic landmasses carried a steady stream of chemicals into this global basin. This primordial ocean was a vast chemical reactor. Into this aqueous environment fell the molecules formed in the atmosphere, along with organic compounds delivered by comets and meteorites. The water itself acted as a universal solvent, allowing these different chemical species to mix, interact, and react with one another. Hydrothermal vents on the ocean floor, spewing superheated mineral-rich fluids from the planet's mantle, would have created localized hotspots of intense chemical activity. It was this unique combination, a reducing atmosphere, an abundance of liquid water, and a constant influx of dissolved chemicals, that created the famous primordial soup, a chemically rich environment where the first steps from geology to biology could be taken. The transformation of simple inorganic chemicals into the complex organic molecules of life is not a spontaneous process. It requires a significant and continuous input of energy. Fortunately, the early Earth was awash with powerful energy sources, turning the entire planet into a dynamic and highly reactive chemical laboratory. The absence of a protective ozone layer meant that the surface was bathed in intense high-energy ultraviolet radiation from the young sun. This UV light carried enough energy to break the chemical bonds of simple molecules like methane, ammonia, and water vapor in the atmosphere, creating highly reactive fragments known as free radicals. These fragments would then recombine in new ways, forming more complex molecules like hydrogen cyanide and formaldehyde, which are key precursors to amino acids and sugars. Another spectacular and ubiquitous source of energy was electricity. The thick, volatile atmosphere, turbulent with heat rising from the surface and constant volcanic eruptions, would have generated immense and perpetual electrical storms. Lightning would have cracked across the skies with a frequency and intensity far beyond anything seen on modern Earth. Each lightning bolt that struck the atmosphere or the ocean surface delivered a powerful jolt of energy, providing the necessary spark to trigger chemical reactions among the atmospheric gases. This process is akin to a natural version of a chemistry flask being zapped with electrodes, a scenario that would later be famously replicated in a laboratory to test the theories of abiogenesis. Furthermore, the planet itself was a tremendous source of geothermal heat. The intense volcanic activity that characterized the Hadean Eon meant that heat from Earth's molten core was constantly being vented to the surface. Lava flows poured across the landscape, and deep-sea hydrothermal vents gushed superheated water, rich in dissolved minerals and gases, into the oceans. These vents created unique environments with steep chemical and thermal gradients, sharp differences in temperature and chemical concentrations over very short distances. Such gradients are powerful engines for chemical reactions, providing both the heat to drive them and the mineral surfaces to catalyze them, making these vents a prime candidate for the location where life may have first begun. Finally, the relentless bombardment by asteroids and comets, while destructive, also contributed a significant amount of energy. The shock waves generated by these massive impacts could create extremely high pressures and temperatures, capable of synthesizing complex organic compounds from simpler precursors. In essence, the early Earth was anything but a tranquil or stable place. It was a violent, energy-saturated world. It was the convergence of all these powerful energy sources, solar radiation, lightning, geothermal heat, and cosmic impacts, acting upon the unique chemical inventory of the atmosphere and oceans, that created the perfect crucible. This was the violent, unstable, and yet chemically perfect laboratory where the building blocks of life could be forged from the raw materials of a young planet. For decades, the idea that life could have emerged from a primordial soup of chemicals on early Earth remained a compelling but honestly purely theoretical concept. Scientists like Alexander Oparin and JBS Haldane had independently proposed that a planet with a reducing atmosphere, liquid water and sufficient energy could spontaneously generate the organic building blocks of life. While this hypothesis was logical and elegant, it really lacked one crucial element: experimental evidence. It was one thing to speculate about the chemical possibilities of a world that existed 4 billion years ago, but it was another thing entirely to prove that such a process could actually happen. The question lingered in the scientific community: could the building blocks of life really be formed so simply, using just the raw materials and conditions of the ancient Earth? This challenge was taken up in the early 1950s by a young graduate student at the University of Chicago named Stanley Miller. He was working under the guidance of his Nobel laureate professor, Harold Urey, a renowned chemist who was deeply intrigued by the chemical origins of the solar system and life itself. Urey had been lecturing on the idea that Earth's early atmosphere was composed of methane, ammonia, hydrogen, and water vapor. A starkly different environment from our current oxygen-rich air. Miller, present at one of these lectures, was captivated by a simple yet profound question. If this was indeed the atmosphere of early Earth, could one recreate it in a laboratory and see if life's fundamental molecules would form? It was a bold and ambitious thought for a student project. Initially, Harold Urey was hesitant. He considered the experiment to be risky and unlikely to yield significant results within the time frame of a doctoral thesis. He worried that his student might spend years on a project that led to a dead end, which is honestly a common fear in the world of pioneering scientific research. He suggested a safer, more conventional project for Miller. However, Stanley Miller's passion and persistence were unwavering. He was convinced that the experiment was not only feasible, but also fundamentally important. He managed to persuade Urey to give him a chance, agreeing that if no significant results were produced within 6 months to a year, he would abandon the project and move on to something else. With Urey's reluctant blessing, Miller set out to design an experiment that would, for the first time, put the Oparin-Haldane hypothesis to a direct and rigorous test. The goal was to build a self-contained, closed system that simulated the proposed conditions of primordial Earth as closely as possible. This was not just a matter of mixing chemicals in a beaker, it was an attempt to reconstruct a miniature, simplified version of an entire planetary environment. He aimed to create a microcosm of a world that no human had ever seen, a world of toxic gases, boiling water, and perpetual lightning, all contained within a sealed loop of glass tubing in a quiet university laboratory. The apparatus designed by Stanley Miller was, honestly, a masterpiece of scientific ingenuity and simplicity. It consisted of a closed loop of sterile glass flasks and tubes, meticulously cleaned to ensure that no contaminating organic molecules from the modern world could interfere with the results. The setup was designed to mimic the cycle of water on a planetary scale. At the bottom of the apparatus, a flask of water was gently heated, representing the primordial ocean. As the water boiled, it produced water vapor, which then traveled up a glass tube and entered a larger 5-liter flask. This upper flask was the heart of the experiment, serving as the simulated atmosphere of the ancient Earth. Into this atmospheric flask, Miller introduced a carefully measured mixture of gases that he and Urey believed represented Earth's early air: methane, ammonia, and hydrogen. This created a miniature, self-contained version of the toxic, oxygen-free atmosphere hypothesized for the Hadean Eon. To simulate the primary energy source that would have driven chemical reactions in this atmosphere, 2 tungsten electrodes were placed inside the flask. These electrodes were connected to a power source that generated continuous high-energy electrical sparks, crackling through the gas mixture. This was Miller's laboratory equivalent of the immense and violent lightning storms that constantly raged across the skies of the young planet. From the atmospheric flask, the gases then passed through a condenser. This was a glass jacket cooled by a continuous flow of cold water, which caused the water vapor in the gas mixture to condense back into liquid droplets, just as water vapor condenses in the atmosphere to form rain. This laboratory rain then dripped down, carrying with it any new chemical compounds that might have been formed in the spark chamber. The liquid collected in a U-shaped trap at the bottom of the apparatus before flowing back into the boiling water flask to begin the cycle anew. This ingenious design ensured that any newly formed, non-volatile molecules would be removed from the spark chamber and protected from being destroyed by the continuous electrical discharge. With the apparatus sealed and the experiment running, all Miller could do was wait and observe. The clear water at the bottom began to boil, the sparks began to crackle, and the cycle of evaporation, reaction, and condensation commenced. For the first day, nothing visibly changed. The water remained clear, and the experiment seemed to be producing nothing of interest. It was a tense period of anticipation, as the success or failure of his bold idea hung in the balance. Miller watched his miniature world churn away, a silent, sterile glass loop holding the potential secrets of our planet's distant past, hoping that chemistry would perform the same remarkable trick it had billions of years prior. As the first day passed with no visible change, a sense of disappointment might have begun to creep in. However, by the second day of the experiment, a subtle but unmistakable transformation began to occur. The clear, colorless water in the collection trap at the bottom of the apparatus started to take on a faint pinkish hue. This was the first sign that new chemical compounds were being synthesized and were accumulating in the system. As the experiment continued to run, the color deepened. Over the course of a week, the solution turned from pink to a dark cloudy red, and eventually to a murky turbid brown. A sticky dark residue began to coat the inside of the flasks. The pristine, sterile water had become a veritable primordial soup. After letting the experiment run for a full week, Miller stopped the process and carefully collected the rich, brown liquid for analysis. The critical moment had arrived. The question was, what exactly had he created? What were the new molecules responsible for the dramatic change in color? Using a technique called paper chromatography, a method for separating and identifying different chemical substances in a mixture, Miller began the painstaking process of analyzing his primordial broth. He had to prove that the new substances were not just random, uninteresting tars, but molecules that held genuine biological significance. The fate of the entire experiment rested on the results of this chemical analysis. The results were nothing short of spectacular and would change the course of origin of life research forever. Miller's analysis revealed that the brown soup contained a variety of complex organic compounds. Most astonishingly, he clearly identified the presence of several amino acids. These were not just any amino acids, they included glycine, alpha-alanine, and beta-alanine, some of the very same molecules that serve as the fundamental building blocks of proteins in every living organism on Earth today. The experiment had succeeded beyond his wildest expectations. In just 1 week, using only simple inorganic gases, water, and energy, he had spontaneously created the essential components of life itself. The publication of these results in the journal Science in 1953 sent shockwaves through the scientific world. It was a stunning vindication of the Oparin-Haldane hypothesis and provided the first powerful, tangible evidence that the transition from simple chemistry to the building blocks of biology was not a fantastical leap, but a plausible and perhaps even straightforward process under the right conditions. Miller had demonstrated that one of the first and most critical steps in abiogenesis could occur naturally. It was a landmark achievement proving that the raw materials for life were not rare or difficult to produce. The experiment showed that life's building blocks can, and likely did, form naturally and abundantly on the early Earth. The Miller-Urey experiment was a watershed moment, transforming the study of the origin of life from a field of philosophical speculation into a legitimate and exciting branch of experimental science. It inspired a generation of chemists and biologists to explore the pathways of prebiotic chemistry, launching countless new experiments that built upon Miller's foundational work. Scientists began experimenting with different gas mixtures, various energy sources like ultraviolet light and shock waves, and other environmental conditions to see what range of organic molecules could be produced. These follow-up studies confirmed and expanded upon Miller's initial findings, showing that a wide array of life's building blocks, including all 20 common amino acids and even the components of nucleotides, could be formed under plausible prebiotic conditions. However, over the decades, our understanding of early Earth's atmosphere has evolved. Newer geological models suggest that the atmosphere may not have been as strongly reducing as Miller and Urey first assumed. It might have been a more neutral atmosphere, dominated by carbon dioxide and nitrogen, with smaller amounts of methane and hydrogen. For a time, critics argued that this change undermined the significance of the original experiment, as neutral atmospheres produce organic molecules less efficiently. This, however, turned out to be a temporary setback. Further research showed that localized environments, such as the areas around volcanic plumes or deep-sea hydrothermal vents, could have maintained the kind of reducing conditions necessary for robust organic synthesis, even if the global atmosphere was more neutral. Remarkably, the story of the Miller-Urey experiment had another chapter waiting to be written, decades after it was first performed. After Stanley Miller's death in 2007, his former student, Jeffrey Botta, discovered a collection of sealed, unanalyzed vials from the original 1950s experiments. Using modern, far more sensitive analytical techniques than were available to Miller, Botta and his team re-examined the contents of these vials. They analyzed samples from a variant of the experiment that simulated a volcanic eruption by adding a jet of steam to the spark chamber. The results, published in 2008, were astounding. They found a much wider variety of organic molecules than Miller had originally reported, identifying over 20 different amino acids, far more than the handful detected in 1953. This posthumous discovery revitalized the legacy of the experiment, demonstrating that volcanic environments on early Earth could have been incredibly potent factories for producing a rich and diverse array of life's building blocks. Today, the Miller-Urey experiment is celebrated not for being a perfect and final depiction of early Earth, but for being the bold, pioneering first step that proved a fundamental principle. It showed humanity that the molecules that make us who we are can arise from the simple laws of chemistry and physics, acting on a barren planet. It was the experiment that took the origin of life out of the realm of pure theory and placed it firmly into the world of testable empirical science, where it continues to be explored today. The successful synthesis of amino acids and other organic molecules in experiments like Miller-Urey's was a monumental achievement. It proved that the universe could readily produce the raw materials for life. However, a collection of loose molecular building blocks, no matter how abundant, is not life itself. The next great challenge on the path to biology was organization and function. In all modern life, these roles are neatly divided. DNA serves as the master blueprint, storing the genetic information, while proteins act as the skilled laborers, carrying out the chemical work. This creates a classic chicken and egg paradox. You can't build proteins without instructions from DNA, but you can't replicate DNA without the help of protein enzymes. So which came first? The RNA world hypothesis offers a brilliant and widely accepted solution to this conundrum. This hypothesis proposes that before the sophisticated DNA-protein system evolved, an earlier, simpler form of life existed based almost entirely on a single remarkable molecule ribonucleic acid, or RNA. This ancient era is imagined as a world where RNA reigns supreme, single-handedly performing the two most critical tasks required for life. Firstly, like its more famous cousin DNA, RNA is a nucleic acid capable of storing genetic information in its sequence of nucleotide bases. It can act as a template, carrying the instructions for its own replication. This ability to store a blueprint is the fundamental basis of heredity, allowing information to be passed down through generations of molecules, a key requirement for any living system. The second, and truly revolutionary aspect of RNA is its ability to act as a catalyst. Unlike the rigid, stable structure of DNA, a single strand of RNA is flexible and can fold back on itself to form intricate and specific three-dimensional shapes. Some of these shapes can function as enzymes, molecules that speed up chemical reactions. These catalytic RNA molecules are called ribozymes. The discovery of ribozymes in the 1980s was a watershed moment, proving that RNA could indeed be both the architect, or information carrier, and the builder, or functional molecule. This dual capacity meant that a single type of molecule could, in theory, run an entire primitive life form, sidestepping the DNA-protein paradox entirely. In this primordial RNA world, the ancient oceans would have been teeming with a diverse population of these versatile RNA strands. These molecules would have been capable of catalyzing their own replication, using the freely available nucleotides in the environment as their raw materials. This self-replication would not have been perfect. Errors or mutations would inevitably occur, creating new RNA sequences with slightly different shapes and properties. This variation was the raw material for a new kind of evolution, one that operated on a purely molecular level. The RNA molecules that were more stable or replicated faster would outcompete their rivals, becoming more abundant over time and setting the stage for increasing complexity. While self-replicating RNA molecules represent a huge leap towards life, A freely floating strand of RNA in the vastness of the primordial ocean faces a critical problem. Its chemical components would immediately drift away and be lost. For a system to truly evolve, it needs a boundary, a container to hold all its essential parts together. This is where lipids come into play. These are fatty, oil-like molecules with a unique relationship with water. One end is attracted to water, or hydrophilic, while the other end is repelled by it, or hydrophobic. When placed in water, they spontaneously self-assemble into microscopic, hollow spheres called micelles or vesicles. They arrange themselves with water-loving heads pointing outwards and water-fearing tails pointing inwards, forming a stable two-layered membrane. This is the blueprint for all cell membranes today. In the early oceans, lipid vesicles were forming all the time. If RNA became trapped inside one of these lipid bubbles, it would represent a monumental step in the origin of life. This primitive structure is known as a protocell. The formation of a protocell conferred enormous advantages. By containing the RNA replicator and its building blocks, replication efficiency would have skyrocketed. The protocell created a concentrated private chemical laboratory where reactions occurred much faster than in the dilute ocean. If the RNA developed new abilities, the benefits would be confined to that specific protocell. The protocell as a whole became the unit of selection. This was the birth of individuality. These early protocells were not yet truly alive, but they were teetering on the very edge. They could grow by absorbing more lipids and could reproduce in a rudimentary way. As a protocell grew larger, it would become unstable and split into smaller daughter vesicles. Each carried precious RNA cargo from the parent. If RNA had replicated before the split, both daughters would inherit genetic information. This simple cycle of growth, replication, and division created a framework for natural selection to act on entire entities. With the emergence of vast populations of protocells, each containing its own set of RNA molecules, the stage was set for the next phase of evolution competition. The resources on early Earth, such as free-floating nucleotides and lipids, were not infinite. Protocells would have been in direct competition with one another for these essential building blocks needed for growth and replication. Those protocells that, by chance, contained RNA molecules that were slightly better at pulling in resources or were more efficient at replicating themselves, would have had a significant survival advantage. They would grow and divide faster than their neighbors, passing on their successful genetic traits to a greater number of offspring. This competitive pressure would have driven a rapid diversification of RNA functions. A protocell containing an RNA ribozyme that could break down other complex molecules would thrive. Another might evolve an RNA that could strengthen its lipid membrane, making it more resilient to environmental stress. This molecular arms race would have favored increasing complexity and efficiency. It was no longer just about which RNA molecule could copy itself the fastest, but which integrated system of RNA and lipids, which protocell, was the most robust and successful reproductive unit. This process actually mirrors the Darwinian evolution we see in the biological world, but played out on a microscopic pre-cellular stage. However, the story was not just one of ruthless competition. Cooperation also would have played a vital role. Inside a single protocell, different RNA molecules could begin to work together, forming the first primitive metabolic pathways. For example, one ribozyme might be skilled at harvesting a simple precursor molecule. A second ribozyme could then take that precursor and convert it into a useful nucleotide. A third could then use that nucleotide to help replicate all the RNA molecules. In this scenario, all three RNA molecules benefit from each other's presence. This kind of synergistic cooperation would have made the protocell far more efficient. These cooperative networks would create a powerful evolutionary feedback loop. Protocells with more complex and effective internal systems would be more successful, and thus natural selection would favor the emergence of these molecular partnerships. Over millions of years, this could have led to incredibly intricate networks of ribozymes, working together to manage energy, synthesize materials, and replicate the entire system. This transition from individual competing molecules to cooperative, integrated systems was a critical step towards the kind of complex chemistry that defines a true living cell. It was the moment chemistry began to behave like biology. The RNA world was a brilliant and necessary stepping stone, but RNA itself has inherent limitations. As a jack of all trades, it is a master of none. While it can store genetic information, its structure is less stable and more prone to mutation than DNA, making it a risky choice for storing a large and complex genetic blueprint. And while it can act as a catalyst, Protein enzymes are far more versatile and orders of magnitude more efficient at speeding up chemical reactions. As protocells grew more complex, there would have been immense selective pressure to develop a more robust and specialized system, to delegate the key tasks of information storage and chemical work to more suitable molecules. The first part of this great molecular takeover likely involved the invention of proteins. At some point, a clever ribozyme within a protocell may have evolved the ability to string together amino acids, which were readily available in the primordial soup. This would have marked the beginning of translation, the process of reading a nucleic acid sequence and turning it into a protein. The first proteins would have been simple, but their catalytic power would have been immense. A protocell that could create even a single type of protein enzyme to assist with replication or metabolism would have instantly gained a massive advantage over its RNA-only competitors, quickly dominating the environment. The second and final major transition was the shift from RNA to DNA as the primary genetic material. DNA, with its stable double helix structure and robust proofreading mechanisms, is a much safer vault for storing precious genetic information over long periods. This transition likely occurred when a new protein enzyme emerged, one called reverse transcriptase, which could read an RNA template and synthesize a more stable DNA copy. Once the genetic information was safely archived in DNA, the RNA molecules were freed from their storage duties. They could now specialize in their modern roles as temporary messengers, transporters, and structural components of the protein-building machinery. With this new division of labor, the modern biological system was born. DNA became the stable, long-term library of genetic information. RNA became the flexible, short-term messenger and multi-purpose tool. And proteins became the powerful and efficient workforce that carried out almost all of the cell's complex chemical functions. This new system was vastly more stable, efficient, and capable of supporting much greater complexity than the old RNA world. The protocell had finally evolved into the first true living cell, complete with a stable genome, a complex metabolism, and the ability to reproduce with high fidelity. This was the last universal common ancestor, or LUCA, from which all subsequent life on Earth would descend. Let's zoom out from the microscopic drama of molecules and protocells to view the grand, sweeping timeline of life's journey on a planetary scale. The emergence of the first true living cell, often referred to as the last universal common ancestor, or LUCA, marked the most significant turning point in Earth's history. This event, which likely occurred around 3.8 to 4 billion years ago, established the foundational operating system for all future biology. This single-celled organism, though infinitesimally small and simple by our standards, was a marvel of cosmic evolution. It possessed a stable DNA genome, a functioning metabolism driven by protein enzymes, and a reliable method of replication, allowing it to begin the chain reaction of life that continues to this day. For an immense period of time, life on Earth remained exclusively microbial. For nearly 2 billion years, almost half of our planet's entire history, these tiny, single-celled organisms were the sole inhabitants of the world. The first cell became 2, then 4, then millions, slowly populating the primordial oceans. These early life forms, known as prokaryotes, which include bacteria and archaea, were incredibly resilient and adaptable. They evolved a diverse array of metabolic strategies to survive in the harsh, oxygen-free environment. Some consumed chemicals bubbling up from hydrothermal vents, while others learned to harness energy from sunlight in a primitive form of photosynthesis that did not produce oxygen. The planet was theirs, a sprawling, unseen empire of microbes. A truly revolutionary innovation occurred around 2.7 billion years ago with the evolution of a new type of bacteria called cyanobacteria. These remarkable microbes developed a new, more efficient form of photosynthesis that used water as a fuel source and, crucially, released oxygen as a waste product. This was a pivotal moment. As these cyanobacteria flourished in the oceans, they began to pump enormous quantities of this highly reactive gas into the atmosphere. For the existing life forms which had evolved in an oxygen-free world, this was a catastrophe. Oxygen was a deadly poison, leading to what is known as the Great Oxidation Event, Earth's first mass extinction, which wiped out a vast number of anaerobic organisms. However, this crisis also created an incredible opportunity. The accumulation of oxygen in the atmosphere fundamentally and permanently transformed the planet's chemistry and climate. It also paved the way for a new, more powerful way to generate energy: aerobic respiration. Some microbes evolved the ability to not only tolerate oxygen, but to use it to extract far more energy from their food than was possible before. This metabolic revolution set the stage for the next great leap in evolutionary history, the emergence of larger, more complex cells. The world, once ruled by simple anaerobes, was now primed for a new kind of life to take over. The rise of oxygen in the atmosphere fueled the evolution of a new and profoundly more complex type of cell. Around 2 billion years ago, the eukaryotic cell emerged. This was a revolutionary step up from the simpler prokaryotic cells that had dominated the planet for so long. Eukaryotic cells are fundamentally different. They are much larger and feature a host of specialized internal compartments called organelles, each with a specific job. Most importantly, they possess a true nucleus, a membrane-bound sac that houses and protects the cell's precious DNA. This new cellular architecture allowed for a much greater degree of complexity and control over a larger genome, opening up a universe of new evolutionary possibilities. The origin of the eukaryotic cell is explained by the theory of endosymbiosis. This theory proposes that the complex cell was born from a partnership, a merger of different prokaryotic organisms. It is thought that a large ancestral host cell engulfed a smaller bacterium capable of aerobic respiration, using oxygen to produce energy. Instead of being digested, this smaller bacterium remained, living and reproducing inside the host. Over millions of years, the two became completely interdependent. The host cell provided protection and nutrients, while the engulfed bacterium became a highly efficient power plant for the cell. This ancient bacterium evolved into what we now know as the mitochondrion, the powerhouse of all complex cells, including our own. A similar endosymbiotic event occurred a second time, when some of these early eukaryotic cells engulfed a photosynthetic cyanobacterium. This captured microbe evolved into the chloroplast, the organelle that allows plants and algae to perform photosynthesis. With these new internal power sources and a highly organized cellular structure, eukaryotes had the energy and genetic capacity to experiment with new forms and functions. For another billion years, life remained predominantly single-celled, but the oceans were now filled with a diverse array of these complex eukaryotic microbes, such as amoebas, algae, and protozoa, competing and coexisting with the still abundant prokaryotes. The final major hurdle before the explosion of visible life was the evolution of multicellularity. This happened not once, but multiple times independently in different eukaryotic lineages. Around 1 billion years ago, some single-celled organisms began to live in colonies. Over time, the cells within these colonies became specialized, taking on different roles, some for movement, others for feeding, and some for reproduction. This division of labor allowed for the development of larger, more complex organisms. The first multicellular life forms were simple, like sponges and primitive algae, but they represented a fundamental shift. Life was no longer just a collection of individual cells. It was now capable of building integrated, coordinated bodies. For billions of years, life on Earth was simple, mostly microscopic and soft-bodied. Then, about 541 million years ago, evolution accelerated in the Cambrian explosion, filling the oceans with complex, bizarre creatures like trilobites and Anomalocaris. Nearly all major animal body plans appeared, setting the stage for the diversity we see today. Rising oxygen, predation, and new genetic toolkits fueled this burst of innovation. From this explosion came fish, amphibians, reptiles, mammals, and eventually us, Homo sapiens. One ancient cell branched into the magnificent tree of life, connecting all living things. Abiogenesis explains how life began on Earth, but the laws behind it are universal. The elements of life, carbon, hydrogen, oxygen, and more, are everywhere, forged in stars and scattered across the cosmos. If the right conditions exist elsewhere, life could emerge on other worlds just as it did here. With billions of planets in our galaxy alone, the odds favor life arising beyond Earth. Our story may be just one example of a cosmic phenomenon, not a unique miracle. We are the universe becoming aware of itself, asking if it's alone. The search for life is now a scientific quest, guided by the principle, follow the water. Moons like Europa and Enceladus with hidden oceans and organic molecules are prime targets. Telescopes scan exoplanets for biosignatures, gases like oxygen and methane that hint at life. Finding even simple microbes elsewhere would prove life is a universal feature, not a fluke. The universe may be teeming with biology, each world writing its own story. As we search, we wonder, are we alone or just one voice in a cosmic chorus? Our atoms were forged in ancient stars, making us literal stardust. Life's emergence is a continuation of cosmic evolution, from chaos to complexity. We are animated chemistry, deeply connected to the universe and Earth's history. Our genes echo a 4 billion-year journey from molecules to conscious beings. This scientific story is as awe-inspiring as any myth, built on curiosity and evidence. We are the universe's way of knowing itself, carrying the torch of wonder forward. The origin of life remains a thrilling mystery, with many questions still unanswered. Our curiosity drives us to explore, experiment, and connect the dots across science. Searching for life elsewhere is a search for ourselves, a quest to see if we're part of something bigger. Even the simplest discovery beyond Earth would reshape our understanding of life's place in the cosmos. The journey from atoms to us is a story of wonder, urging us to stay curious and keep exploring. The adventure continues. Stay curious, stay wondering, stay alive.